Hello, everybody, and welcome to Meiosis. So we just finished up mitosis. I graded a lot of tests, and people did very well. So hopefully we're watching the videos, and we're taking the notes seriously, and we're taking the project seriously. And with that, we should be all set. Now, meiosis is not that much more difficult. I mean, it's way more difficult, actually, but I'm not going to make it that much more difficult than mitosis. But you just have to pay attention to the differences. And as long as you pay attention to the differences and where these cells take place, things will be great. So let's see what we got. Meiosis, instead of having 2 daughter cells, in meiosis, you're going to have 4. Now, if we remember in mitosis, we have 2 cells that had the exact number of chromosomes, and they were identical to each other in every aspect. Here, you're going to end up with half of the chromosomes, and each one is going to be genetically different. So basically, meiosis is nothing like mitosis. Well, it is because there's cell division, but there's a lot of differences. So as long as you pay attention to the differences, the questions will be a piece of cake. So if you look here, the original cell over here has a number of 4 chromosomes. And you can see that. You got 1, 2, 3, 4. After meiosis 1, well, actually, meiosis 1 is going to be over here. So there's going to be 2 rounds. There's going to be meiosis 1, and there's going to be meiosis 2. Now, there's way more to the story than this, but what's going to happen is, just like in mitosis, there will be DNA replication. So if you notice here, you go from 4 single-stranded chromosomes to 4 double-stranded chromosomes. You did not add any chromosomes, you simply doubled your amount of DNA. After meiosis 1, you end up with 2 cells that have these chromosomes that look like they've swapped some material right there, and we'll talk about that in a little bit. And then after meiosis 2 is done, you end up with 4 cells, but they have half of the chromosomes. See, look, only 2 and 2 and 2 and 2, and you started with 4. And if you notice, you had, let's say, a long blue and a long red. Those are homologous. Then you had a short blue and a short red, and those are homologous. Well, over here, you can see you have a mostly long red with a little bit of blue and a short blue. Here you have a totally long red with a short blue with a little bit of red, long blue, short red with a little bit of blue, short red, long blue with a little bit of red. Don't get confused in the colors. Just see that now each of those cells is genetically different. So this is a nice summary of what the last slide just said. 4 cells with half of the chromosomes that you started with, and they're all genetically different. Gametes, that's what we're creating here. In the male, we're going to call them sperm, and in the female, we're going to call them eggs. Words that I'm sure you have heard before. The egg is very large compared to the sperm. The sperm is very small. So what's going to happen is you're going to find out that males produce lots and lots and lots and lots of sperm, like millions and millions and millions every month. And females will mature 1 egg. Crazy, right? So you may note the egg is also called an ovum, and the sperm is also called a spermatozoa. So you may see that. The gonads are where these gametes live. So in the males, the gonad is the testes. That's where the sperm is. And in the ladies, the gonads are the ovaries, and that's where your eggs are. The ovaries will release the egg. We'll talk more about that in the reproductive system. And the testes will release the sperm, which again we'll talk more about in the reproductive system. This is just a quick overview. Don't spend too much time on this now because we're going to spend a lot more on it when we do the reproductive system. But there's your testes, and ladies, your ovaries are right there. So again, we'll see way more when we get to that in the reproductive system. So let's start with spermatogenesis. That is the formation of sperm. Now, what makes this different is men are going to start out with something called a spermatogonium. The ladies will be called an oogonium. That is a diploid cell, which is basically has 2 copies of each chromosome, just like your regular cells. But these guys are a little different, so don't think they're the same. And what you're going to end up with is 4 genetically different cells, and I'm going to link that with the word haploid. Haploid means half of the chromosomes, or they have only 1 of each chromosome. And we'll talk more about that as we go. The sperm are going to have a tail. Female eggs do not have a tail. And males can produce 60,000 of them per minute. So males, you are making a lot of sperm. So 4 genetically different cells. They're all viable. What that means is for every 1 spermatogonium a male has, 4 genetically different sperm, each is viable. Each will work. We're going to see that in the females. That's not the case. Again, if you look here, you start off with your spermatogonium, which is a diploid cell, which has 2N, which means it has 2 copies of each chromosome. By the time you're done, you have the haploid N, and these guys are all genetically different, and they all have a tail. You will see here that the replication will happen right there, and then you will have division number 1, and then you will have division number 2. So meiosis is 1 replication, 2 divisions. Oogenesis is the formation of the eggs, which happens in the ovaries. Females, instead of starting with a spermatogonium, are going to start with an oogonium. Now here's what happens. Same deal. By the time you're done, you're going to end up with 4 cells. But here you're going to end up with 1 egg and 3 polar bodies. Now keep in mind that in meiosis, you always have to end up with 4 cells. I'm not saying all 4 cells are going to be viable. In this case, only 1 is. So you produce 1 haploid egg and 3 polar bodies. This is per month. Males are going to make thousands and thousands and thousands a minute. Females are going to make 1 per month. Some women will release 2, and then you'll have fraternal twins, which we'll again talk about in the reproductive system. But I just wanted you to see here that the female egg is big because it has all of the organelles that you're going to need. And we remember from last chapter, organelles like ribosomes and endoplasmic reticulum and mitochondria and all that good stuff. So remember, the egg is huge. Make 1 per month, and it has all of the organelles that you need. So again, here's your 3 polar bodies, 1, 2, 3. These in essence cannot go on to make a baby. The egg or the ovum can, and you will notice how much bigger it is. That's again what kind of leads to some genetic diversity. Once again, your 4 cells here, notice you have 4 chromosomes. Here you have only 2. You replicated right there. Just like in mitosis, there's split 1, there's split 2. 1 replication, 2 divisions. It's all about the numbers. Your cells, which are known as your somatic cells, all have 46 chromosomes. Yours, humans. The oogonium and spermatogonium also have 46 chromosomes. But by the time you get to these sperm and egg, they only have 23. And the reason why is the 46 chromosomes that you have, you got 23 from mom's egg, and you got 23 from dad's sperm. That's where your number comes from. So keep that in mind. When you see the haploid number, once again, that's going to be the N number or the half number found in the gametes. And when you see this word diploid, that's going to be the full set of chromosomes. And what I mean by full set is you have 46 chromosomes or 23 pairs, which means you got 23 from mom and 23 from dad. So this here is a karyotype. This is a normal karyotype, and you know it's normal because if you count it up, There are 46 chromosomes. You see there are 23 numbers. Now this X and Y, that's chromosome number 23, otherwise known as your sex chromosome. The reason why they break it up is to see if you're a boy or a girl. There are 2 X chromosomes here, so that means this is a female. And if this is chromosome number 1, you got that one, let's say, from mom, and you got that one from your dad. And there's your homologous chromosomes. This is your diploid set. You have 2 of each chromosome for a total of 46. Or in humans, this is your 2n number because you have 23 numbered chromosomes. See, 1, 2, 3, all the way up to this XY, which is number 23. That's your n, but you got 2 of them, one from each parent. So that's your 2n number. Once again, this is a normal karyotype. And some chromosomes are bigger than others, but that's okay. Homologous chromosomes mean you got one from your mom. Let's say that this is mom's chromosome 1 and this is dad's chromosome 1. They're going to be the same length, so that's good. They're going to have the same gene. So for example, let's say the eye color gene is here. Well, the eye color gene is going to be there on dad. Now, they are not identical. They're not exact copies of each other, but they are the same in that they will have the genes located in the same spot. They're not identical. Of course, they're not identical, but the genes are located in the same spot. So this here is a fancy color version, let's just say. So if this is mom's 7, let's just say, that means this is dad's 7. And you can see that the gene for C, whatever C is there. And the gene for B is right there, whatever that is. And A is right there. They cannot be flip-flopped all willy-nilly. It doesn't work that way. Homologous means they're the same number, but they're from different parents. So you can see here, once again, the green and the purple are homologous. Again, let's say that this is, I don't know, mom's 6, which makes this dad's 6. After replication, here you had 2 single-stranded chromosomes. Now you have 2 double-stranded chromosomes. You did not add chromosomes, you added DNA. Now, we talked about this last time. These guys are called sister chromatids. These guys are exact to each other, and these guys are exact to each other, because eventually there's going to be a little swapping going on and all this stuff. But again, homologous means one of each. You can see that's pointing to green and to purple, and the sisters are linked by this thing called a centromere. But we learned that last time. So there's slight confusion here, which makes things a little bit tricky. In meiosis, just like I said a couple times, the chromosomes are going to replicate once. That's going to make your sisters, but they're going to split twice, and that's what's going to take you to your ultimate 4 cells. The replicated chromosomes, once again, are called sister chromatids, which we just saw in the picture. And what's going to happen is they're going to do a little interacting. They're going to do a little swapping of material. That's why it is important that the genes are in the same spot. So let's see what I'm talking about. Under normal circumstances, let's say, for example, this cell, you know that this is interphase. And it's interphase because the DNA is all squiggly squaggly. You know here that the centrioles are kind of bunched up. I don't see any spindle fibers. I see my nuclear membrane is intact. Put a little M on there. And I know everything's cool. This is just a regular cell doing regular cell things. But watch what's going to happen. Still an interphase. This is now the S phase, because look what happened. You went from 1 blue to 2 blues, 1 red to 2 reds, 1 green to 2 greens, and 1 purple to 2 purples. Notice you're still in interphase, the S phase, because these guys have not moved, and I do not see any spindle fibers, and my nucleus is still there. So far, we did our only replication. That's all we did. So what do we got? Meiosis 1 is 4 phases, then it splits, and meiosis 2 is going to have the same thing. I put the asterisk here on prophase 1 because that's the big deal. this is going to get a little love and this is going to get a little love. But meiosis, prophase 1 is a big deal. So prophase, just like in mitosis, nuclear envelope, bye-bye. Nucleolus, bye-bye. Centrioles start to move, sounds good. Spindle fibers form, good. And the chromosomes are going to condense. All right, so far so good. Nothing is different from mitosis, except, ding, 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 that. During prophase 1, what's going to happen is these homologous pairs are now known as tetrads. And the reason why they're tetrads is if that's mom's chromosome 1, that's the copy of mom's chromosome 1. That's dad's chromosome 1. That's the copy of dad's chromosome 1. They're called tetrads because there's a total of 4 of them. And what's going to happen is there's going to be something called crossing over at this place called the chiasma. So let me show you what happens. Everything's cool there. We know we're in prophase 1 because the chromosomes have condensed nicely. We see that our centrioles are starting to move. We have our spindle fibers, which are right there, and our nucleus is starting to dissolve. So we're in prophase 1, just like in mitosis. But here's where things get a little tricky. Remember, homologous chromosomes mean they're the same number. They have the same genes in the same spot. They're not identical, but the genes are in the same spot. So once again, let's say that the gene down here, whatever, or the gene up here, they're the same gene. They have to be held together at a centromere. They're called tetrads, once again, because there's a total of 4 sisters. 1, 2, 3, 4. There's your tetra or tetrad. Well, something is crazy going to happen in prophase 1 at this thing called the chiasma. What's going to happen is there's going to be an interaction. Now, what that means is the sisters of homologous pairs are going to interact. Now, remember, I told you it was very important that the genes are in the same spot because what they're ultimately going to do here is they're going to do the old switcheroo. So watch carefully. See how they did a little swap of just a piece? Now, they didn't swap too much, but they swapped just the end. And the act of switching up the parts of the homologous chromosomes is known as crossing over. This is what is ultimately going to lead to that genetic diversity. So if you remember in that very first picture that I showed you, when the slides were those 4 circles all the way on the right, they were mixed and matched, all because of crossing over. There it is in the kind of picture scene of it. Now, metaphase is going to be a little different. Now, if you remember in mitosis, when I drew the metaphase, I had all the paired chromosomes, and they were all lined up down the middle, sort of kind of like a belt, just like that. But in metaphase 1, they're going to line up like this, where the pairs of these chromosomes are going to be next to each other. So it's kind of obvious now to see that these are not the same orientation, because where in mitosis I simply split the sisters and everything was fine. That's what I did in mitosis, if you remember. But in meiosis, what's going to happen is this whole package deal is going to go that way, and this whole package deal is going to go that way. So watch. There they are. They're not on top of each other. They are now all next to each other. And what is going to happen is the whole pair is going to move. So that's going to move, and that's going to move together. We don't split the sisters yet. So one of the questions on your notes is how is metaphase different? In metaphase, in meiosis, the homologous chromosomes are next to each other, not lined up on top of each other. In anaphase, note the homologous pairs are separated, not the sister chromatids. If you remember, in mitosis, we split the sisters. Here we're taking the whole pair, which I showed you before. So if you look, this is now different. So how is anaphase different? Instead of splitting the sister chromatids, you take the whole homologous pair with you. Now that's going to lead to an interesting scenario. By the time telophase 1/cytokinesis is over, you're going to end up with 2 cells. Now this is where things get a little tricky, and again, I'm not going to hold you crazy to this. But the cells have the same amount of DNA that they started with. You're like, what? Because let's look. If you remember, I started with 2 long chromosomes and 2 short chromosomes. Well, after I split this, I'm still going to have 2 long and 2 short. They don't look the same, but it's the same amount of DNA, and that's where kids get a little tripped up on this. The cells are not identical. They're not even close to identical, but I have the same amount of DNA than when I started. So now I'm going to begin meiosis 2. Now what meiosis 2 is ultimately going to do is it's ultimately going to split these guys up like that. Now notice in meiosis 2, there is no replication. All I'm going to do is break them up. So meiosis 2, the sister chromatids are now going to be separated. So let's kind of see how this goes. And there is not a second round of replication. This is division only. So let's see what happens. There they are. These are the cells after meiosis 1. And now look what happened. I didn't replicate anything. Now I just divided it. So if you look, I started with 4 chromosomes, if you remember. Now I have 4 cells where each has only 2 chromosomes. So therefore, I split my amount in half. And if you notice, none of them are the same. They are all genetically different. That's why you may or may not look like your siblings. You may look nothing like your siblings. It's all due to random chance of meiosis. So it's all about genetic diversity, and you can see here that they're all just slight bit different. So this one here has a complete long blue one. This one has blue with a little bit of red. This one's got red with a little bit of blue, and this one's got all red. And if you look at the purple and the green, the same thing. So they're each slightly different to the one next to them. That is genetic diversity. So let's review. Let's see if it all sounds good. There's our 4 chromosomes that we started with. Here we know this is replication. This is meiosis 1. This is meiosis 2. Notice you end up with 4 cells that have half of the amount of DNA you started with, and each one is genetically different. So if you get that straight, the questions will be pretty easy. Well, what happens if there's a mistake? In mitosis, we know that each of the new cells will have the total wrong number of chromosomes and things are bad. But in the world of meiosis, there's a couple of times when you might actually get away with making a new baby. And it's called non-disjunction. Now what happens here is instead of having four, why is it doing that? Instead of having 4 cells that have an equal number of chromosomes, you can see here how this one kind of double grabbed, which left this blank. Now, if you leave that blank, you're going to have too many over here, and here you're going to have not enough. Now, in some cases, this will not lead to proper fertilization, and the baby will never be born. But in the case of Turner syndrome or Klinefelter syndrome or Down syndrome, the baby will survive. So it's all about the slight difference in the number of chromosomes. So here you can see that non-disjunction can happen in 2 spots. If it happens early enough, like after meiosis 1, then you got nothing over here. If it happens after meiosis 2, where most of the surviving babies this will happen, you will see here that normal means, let's say we're supposed to have 1 chromosome each. Here there's none, and here there are 2. So this is going to lead to extra. This is going to lead to not enough. And let's see what happens. Oh, this is a review again. So if all is good, you started off, and you're supposed to have nice 4 cells, even. Again, here you got 2 mistakes, 2 good. And here you got all bad. So if you notice, depending on where the non-disjunction takes place, will determine how many cells are affected. And in this case, here's the problem. If, let's say, dad brought an extra and mom brought the normal amount, you're going to have a kid with an extra chromosome. So in the case of Down syndrome, they have an extra chromosome number 23. And I'll show you a karyotype in a second. So if you notice here, chromosome 1's fine. They got 2 of each. Chromosome 2 is fine. Everything's cool. This is a boy, and you know that because you see the Y chromosome. But this is a boy with Down syndrome because if you notice, they have an extra chromosome at spot number 21. That could have come from mom or that could have come from dad. And we will talk more about that in genetic disorders. So that's meiosis. You got questions that will accompany it. I'll give you some multiples to practice. There'll be a project and a test somewhere, probably at the end of next week. Okay? So hopefully that was helpful.